Hello everyone, welcome back to another class on regeneration biology. In today's class, we will learn about how stem cells are tweaked using or how stem cells are modified using CRISPR-Cas9 technology for modifying their genome. So many of you would have heard about it, CRISPR-Cas9 technology. I think in the year 2021, two women scientists received a Nobel Prize for CRISPR-Cas9 technology. And I also have written one article in Resonance about it. Those who are interested can read that article on CRISPR-Cas9 technique. So let us have an overview of the CRISPR-Cas9 technology. Induced pluripotent stem cells can differentiate into specific cell types or tissues of the eye or any other organ or tissues of in humans. You can make them whatever you want, and they have several application prospects in retinal cell transplantation, corneal cell transplantation, and lens regeneration. But what is important we should understand is if a given cell type is not functioning in a tissue or organ, say retina, it could be because of a genetic mutation, maybe in that given cell or maybe in its progenitor itself. That is why the gene editing comes into picture. Gene editing technology of clustered, regularly interspaced short palindromic repeats, that is the short form of CRISPR, and also the CRISPR associated protein 9 known as Cas9, which is basically a nuclease, and short form they are known as CRISPR-Cas9, can effectively introduce causative mutations of inherited ocular diseases into human induced pluripotent stem cells. Means you can do in both ways. Say you have a normal stem cell and you wanted to study the effect of mutation on a given cell type. So what you have to do is have the stem cell, introduce this mutation, and then differentiate into a given cell type and study its physiology, that cellular physiology. Other way around, you have a patient who have a mutation in a given gene, hence their organ is suffering. Collect the cell, of course that is a mutated gene bearing cell, but you correct it using CRISPR-Cas9 technology and now differentiate back into a normal cell, or which is now lacking this defect. So both ways it's possible. Then they can be further differentiated into specific somatic cells maintaining a genetic disease background to, just like I told you, you introduce a mutation and study, which can mimic the occurrence of inherited ocular diseases in vitro. And if it's an animal model, you can study in vivo as well. The cell model may help scientists to study the mechanism of human disease development, and you can establish an in vitro screening platform to find therapeutic drugs and correct genetic mutations in the human genome for cell therapy. So you can study, oh, there is a defect, I have hundreds of drugs, and see how the cell is going to behave. Can one of these drugs can be used for fixing this trouble? One approach. Another approach is, oh, there is a mutation, a given mutation in a patient, and can I fix it and get a normally functioning or a normal cell out of or normally performing cell out of this mutated cell by CRISPR-Cas9 technology. Combining stem cells and gene editing technology revolutionizes regenerative medicine in ophthalmology, and gene therapy of inherited ocular diseases also made possible. Why CRISPR-Cas9? One can always ask, is there no other approach? Of course, there are different approaches are there, and CRISPR-Cas9 is so attractive because it's very, very easy. It is time saver. It's a big time saver. The advent of CRISPR-Cas9 possibly the biggest advance in biological science since the generation of iPSCs themselves. So if you ask someone, ask what is the most beneficial thing that has come ever since the discovery of induced pluripotent stem cell, one must say it is the CRISPR-Cas9 technology that can fix any trouble. So stem cell is a one big achievement, but if there is a trouble or if you want to create a trouble, CRISPR-Cas9 becomes the most attractive choice. And it has opened the doors before only dreamed of accessible genetically engineered patient specific stem cells on demand. Now you can whatever mutation you want, whatever change you want in the genome of an organism, you can make it in no time. Latest advancement of CRISPR-Cas9 has enabled the genome and epigenome editing as well as gene repression and activation. So far, we know the CRISPR-Cas9 is basically causing a cut, a nick in the DNA and removes, or you can, after making a nick, you can fix it by recombination. Now that is not it. You can use Cas9 as a molecule, not as a nicking enzyme, but rather it can act as an activator of transcription or repressor of transcription. Say a person is having cancer because a gene is activated. You can use appropriate CRISPR technology and suppress that gene so that there will not be any more that gene product being formed. So that is one of the other lines of growth that has happened in the CRISPR-Cas9 approach. So the CRISPR-Cas9 approach, the function is very simple. The mechanism of action of CRISPR-Cas9, the gRNA interacts, a guide RNA, it is called, the gRNA means guide RNA, interacts with the Cas9 to recognize 20 nucleotide target sequence located upstream of the invariant 3 base pair region known as NGG. NGG, N stands for any nucleotide and GG. That is the sequence that will be recognized by the Cas9, and that's known as PAM sequence. Photospacer adjacent motif, that is what it is called, sequence, which cause a double strand break in the specific genomic region. Subsequently, DNA repair systems correct the gap in the sequence either by non-homologous end joining known as NHEJ or HDR pathway. That is, you have to have a donor cassette that is present, that is by recombination occurs from a donor DNA cassette and it gets integrated. So you can see here, this is a DNA and you have got a guide RNA that has bound to the one of the loci, which has got Cas9 that is bound onto it, and it is going to create in the NGG region, it is going to create a cut, and it created a double stranded break. And the double stranded break, once it has occurred, the system will try to repair. Repair can be NHEJ, where you can have a random sequence that will be inserted. Because of this, if the cut happened inside a gene, most likely that gene has lost its function. Either it will create a stop codon or it will create a frame shift. Less likely that it will restore the normalcy of the gene. Another option is the precise DNA replacement. So whenever there is a DNA damage, the system or the cell look for a complementary sequence which can integrate by homologous end joining. That means it will recombine. As you can see here, this is the HDR sequence which recombines, as you can see here, and you end up getting a insertion, and now the whole region got fixed. And when you think about, like I mentioned, that you can disrupt, you can create a precise mutation, you can have that precise mutation in this HDR region, create a nick and then insert that HDR cassette with a precise mutation, one option. Or other option is, if you simply want to disrupt a gene and then want to know what is its effect, you can go for NHEJ method. So in either way, it is a very effective and powerful technique. Application of CRISPR-Cas9 in HiPSC, which means human induced pluripotent stem cells. Okay, so that is HiPSC means. Since the initial reports of Yamanaka and colleagues of generating iPSCs in 2007, human iPSCs have been successfully generated by ectopic expression of 4 transcription factor KLF, OCT4, SOX2 and CMYC. So subsequently, several improvements in the method of iPSC generation were reported, including the generation of human iPSCs without overexpression of the oncogenic gene MYC. Because although MYC is one of the Yamanaka factors, MYC always brings in some extra baggage that it can make the cells, the iPSCs, more tumorogenic. So reprogramming methods with mRNA, protein, small molecules, microRNAs, and all of which do not require genomic integration of the reprogramming factors were also devised. Without any too much of complication, you can simply make a stem cell without manipulating the genome. You can do with various other molecules such as drug or a protein or microRNA, etc. The derivation and the culture of HIPSCs is in completely defined xeno-free condition. Xeno means strange material. Xeno-free condition have also paved the way to employ these cells in basic and translational applications. The technological advances have allowed the development of HIPSCs based models on numerous diseases. However, we should understand that full realization of the potential of these cells in research and clinical endeavors definitely require the ability to modify their genome at precise locations. That is what CRISPR-Cas9 offers. CRISPR-Cas9 to generate HIPSC reporter lines to study the cell fate. One approach we will study. The ability to insert reporter genes to the specific genomic loci, say GFP, say RFP, or any other reporter molecule, so that you can know if you want to study the expression of a given gene, you simply integrate a GFP downstream of that gene so that the gene is not affected, but you will be able to see in vitro or in a live condition, if it is a animal that can be put under the microscope, that you can see the GFP expression. To specific genomic loci in HIPSCs offers the chance to faithfully reproduce gene expression patterns with the minimal effect on the adjacent genes. That is the beauty of a CRISPR-based integration of an expression reporter line or an expression cassette. Several studies reported using CRISPR-Cas9 technologies to insert fluorescence protein into loci associated with the undifferentiated state of HIPSCs. Say for example, let us think of a scenario. I have a cell, I took the fibroblast and I am trying to make it stem cell. Stem cells have to have some markers, but who will come and tell me it became stem cell? So my goal is to identify when a stem cell marker start expressing. So if that stem cell marker is now tagged with GFP, whenever I convert a fibroblast into stem cell, I will be able to recognize it by GFP expression, and I can handpick those cells and make a new colony or new passage. For example, green fluorescent protein was inserted downstream of the last exon of the OCT4 gene. OCT4 is a stem cell marker. Generation of HIPSCs reporter line not only could recapitulate OCT4 expression to monitor pluripotency, but they also did not impact the ability of the targeted cells to differentiate into other cell types. Means this GFP integration did not affect the functionality of OCT4 and the functionality of the stem cells itself. That means it's a completely harmless approach. Most importantly, the generation of tissue-specific reporter iPSC lines provides a better understanding of the developmental stage of these cells. Where it is now you have a strong GFP, that means the OCT4 expression is really strong. Or a weak GFP means OCT4 expression is really weak. So sometimes strong expression may be harmful, sometimes weak expression may be useless. So you will be able to catch which is the ideally expressing, just like putting salt in a dish. Too much salt is problem, too less salt is problem. So that also possible by monitoring these reporter lines. We will see few examples. Wu et al. Generated a knock-in mouse iPSC line with the early myogenic lineage specification of a gene called PAX7 using the CRISPR-Cas9 nickase pairs. A GFP reporter with the selection markers was incorporated before the stop codon of the PAX7 gene at the last exon. That means PAX7 gene is intact, you put an extra GFP into it. A similar example of reporter line was generated in human iPSC line where an early myogenic specification gene MIF5 was generated by CRISPR double nickase based knock-in of GFP. So these putting a reporter line is normally referred to as knock-in lines just before the stop codon, which allowed the prospective identification and purification of myogenic progenitors from human iPSC cells. So strategies such as these could improve our ability to track the survival, engraftment and localization of human iPSC derived cell populations in real time in vivo. That means within the organism itself, which is very important for their clinical translation. Without seeing is believing. Like without seeing, you don't know which cell is gone where and what it is doing. So using CRISPR-Cas9 in producing human iPSCs based disease models. Let us see some example. Compared with the conventional stem cell manipulation such as the HDR system for gene modification, CRISPR-Cas9 mediated gene editing has dramatically improved the gene editing feasibility by reducing the time required to recover the edited clones from years to months. Earlier it used to take 4 years, 5 years like that. Now you can do in a matter of few months. It is so easy that if you want a modification in the genome using CRISPR-Cas9 approach. Several patient-derived iPSC lines have been easily engineered via CRISPR-Cas9 to repair the disease-causing mutations, which I already told you. And these were then differentiated and transplanted into the appropriate organ in an animal model, leading to the functional rescue. Let us see a few more examples. Several other researchers have also reported CRISPR-Cas9 system which was successfully used to induce large gene deletions to remove the mutation hotspot in human Duchenne muscular dystrophy using the patient-derived iPSCs. So, in a simplistic sense, Duchenne muscular dystrophy is a male-specific gene. Females can be carriers, and the victims normally don't live beyond 30 years. Some exceptions may be there because their muscles will be very weak, their breathing will get affected, lungs will not function. So these are all very serious issues. But the problematic region itself, you can delete it because Duchenne muscular dystrophy, the dystrophin gene is a large gene. If I'm not wrong, it's around 17 kb RNA itself, 17,000 bases. So this approach may potentially treat 60% of the DMD population. So if there is a problematic area, delete that problematic area so that no more mutations stay in that protein. Hence, the organism survive perfectly. And more importantly, the iPSC correction and the restoration of function have also been applied for the most common chronic lung disease known as cystic fibrosis. We would have heard about it because which affects the chloride balance in the lung epithelial cells. This disease which is caused by a cystic fibrosis transmembrane receptor, also known as CFTR, and which the mutation on this CFTR gene is the main cause of cystic fibrosis disease. Patient-derived iPSCs used to model the correction of the CFTR mutation by transfection with Cas9 and sgRNA plasmid. You put in together and the HDR can recombine and fix the mutation. These cells were subsequently differentiated into mature airway epithelial cells with recovery of CFTR expression. So you can make them into cells of the airway epithelium and you can transfer. And then transplanted, leading to the improved chloride current in response to the stimulation and the disease amelioration. So the complexity of the disease is controlled and the patient survives. So if you see further the application of CRISPR-Cas9 systems in human iPSC, this picture taken from these publications, you can see human and the patient-derived iPSCs are generated, and now you are culturing them and culturing after editing. So first you culture these cells, edit using the CRISPR-Cas9 approach, and you grow the edited cells. That means the mutation is now removed and you can allow them to differentiate, direct differentiation into tissues of your interest. And if you have a reporter line, it is much easy, because if it is a stem cell specific reporter line, then once it is differentiated, it will go off. What if you have put a reporter in a differentiation specific line? Then when the stem cell differentiate into that cell type, then the reporter comes up. Either way you can do it. So treatment also can be done. If you have a culture, you can treat with certain drugs and see that when this cell behaves properly. And also several genetic diseases can be corrected in this approach, and mutation can be reversed back to normalcy, and you can implement or implant into the patient. And also surgically transplantation of the affected area can also be done effectively using these cells. So novel CRISPR based genome engineering tools and that can also be applied in the iPSCs. novel tools. What, like I mentioned in the beginning of the talk, not just cutting the genome. You can edit it. That is a very powerful technique. But you don't want to edit because you want to activate some gene. So you cannot say a gene is turned off in a given cell or a given organism. So it is nothing to do with any mutation. Gene is perfectly fine, but it is not turning on. Or a gene is turned on strongly, and because of that some complexity. So you don't want to turn that gene off, or you don't want to create a mutation in that gene. You want to reduce the expressivity of that gene. So two scenario: turning on or increase its level, or turn off or decrease its level. So CRISPR-Cas9 applications now go far beyond DNA editing. Targeted mutation made onto the Cas9 protein on its catalytic site. Catalytic site means it will cause the cutting. And the fusion of Cas9 to diverse effector domains widened the range of genomic alterations possible in iPSCs and other cell types. It is now possible to precisely downregulate or activate genes without introducing changes in the DNA sequence or using novel by the use of novel CRISPR tools. DNA integrity preservation is required for clinical applications of iPSCs, and CRISPR gene regulation can potentially be applied to generate genetically engineered, safer, and genomically stable cells. So by using this kind of activation repression approach, you can say you did not modify the genome at all. You just made it little active. You just made it little low. Just like, you are tweaking the system. Let us see an approach. CRISPR interference known as CRISPR-I or CRISPR-I. Is a CRISPR activation is known as CRISPR-A, or CRISPR-I is interference, CRISPR-A is activation. So are the gene expression modulation systems that enable the expression or activation of genes. Either you can do repression or the interference, or activation or upregulation. Both the systems are based on mutated Cas9 protein called nuclease deactivated Cas9, also known as dCas9. Mutations on the RUVC and HNH domains abolish its DNA cutting abilities while maintaining unaltered DNA binding domains. It will still bind to the DNA, but it do not cut the DNA. dCas9 recognize the target sequence in the same manner as the wild type Cas9. No change in that. However, disrupting the nuclease activity of Cas9 transforms this protein into a generic RNA guided DNA binding protein. It just binds to the DNA. The fusion of dCas9 with effectors converts dCas9 into a programmable transcription factor that can be engineered to target and recruit other transcription factors to any DNA region specified by the gRNA, because you can target any region. There is no region is immune to CRISPR-Cas9 approach, creating the possibility to modulate gene expression in a very specific and versatile manner. So this is the picture that shows deactivated Cas9 and associated proteins actions used to repress and induce gene expression. So you can see two examples. Left hand side is CRISPR-I, right hand side is CRISPR-A. Interference, activation. Concept is same, that you have the DNA and the guide RNA comes and recognize the palm sequence. Cas9 binds, but this Cas9 is deactivated Cas9, dCas9, hence it will not cut the DNA. Now this dCas9 is now fused with a interfering, say like a gene called CRAB, which has been put in there and which will prevent the gene expression. The gene expression which is normally turned on, now turned off. And other option is that you can bring in something called DNA-MT3, DNA methyltransferase. And Cas9 fused with the DNA methyltransferase will methylate those DNA so that the gene expression can be turned off. Other option of activation is that Cas9 now fused with VP64, which is an activator protein. Or other gene is Cas9 is now fused with the TET1, 1011 translocase, which will demethylate the DNA. So both will turn on the gene. So this is what easily can be achieved using CRISPR approach. So editing the epigenome, the complexity of mammalian gene expression regulations opened many avenues for manipulation. On the other hand, some iPSCs applications could benefit more from stable gene regulation, which can be achieved either by gene knockouts or more recently by sequence preserving epigenome editing. So these are all the approaches we can take. DNA methylation in CPG islands is known as repressive mechanism of gene expression. We all know that CPG islands, if methylated, gene expression will not happen. Association of Cas9 with the catalytic domain of DNA MT3 can promote de novo methylation. That means normally it's not methylated, but you can bring methylation on these loci of genes in mouse embryonic stem cells. When Cas9 DNA MT3 was targeted against an active housekeeping gene, say GAPDH, there was a transcription inhibition showing the feasibility of this strategy to be used in stem cell models. Means any gene you can turn it off as per your will. Similar to adding methyl groups to induce gene silencing, removing such as repressive marks has been performed to activate the gene expression by CRISPR-A. This strategy has used for the promoter region of BNDF gene, where Cas9 fused with the catalytic domain TET1. Like I told you, TET1 is 1011 translocase, which causes the demethylation of the CPG island, led to the activation of gene expression in mouse ESCs. So once you remove the methyl group, you can turn the gene on. TET1 catalyzed the conversion of 5-methyl cytosine into 5-hydroxymethyl cytosine, playing a key role in the active DNA demethylation. Moreover, the Cas9 TET1 was used against a transcriptionally inactivated gene, DAZL. It's a off gene, turned off gene. Transcription was activated, demonstrating the predictability of the system in modulating the gene expression without altering the DNA sequence and with minimal off-target effects. So the researchers have also shown that this system worked in vivo by demethylation of an imprinted gene. Imprinted gene, we will not go into the details. You can read about imprinting. It's a very important step in the fixing of paternal and maternal copy of the gene. So these findings are remarkable for opening the possibility of activation of the endogenous genes in a controllable and inducible manner. So we should understand from this study that even imprinted genes, very quickly I will explain imprinted because you will understand the depth of it. Sometimes say human insulin-like growth factor, you have copy from father and mother of both genes, but the insulin-like growth factor receptor will work only from mother. Father has the gene, but it will not work at all. Permanently it is turned off. Same way, insulin-like growth factor, it will work only from paternal copy. Maternal copy is fine, it's there, but it will not express. This is called imprinting. So sometimes what happens if the paternal copy has got a mutation, maternal copy is very much there, but it has a mutation and now you are suffering. Now the maternal copy can be activated and you will get an extra life. So this is the application. So you can reverse the imprinting. So to conclude, the crosstalk between stem cells and CRISPR technology is improving day by day. While human iPSCs provide an infinite supply of cells for the manipulation of phenotypes and differentiation into tissues for disease modeling and possible therapies, CRISPR-Cas9 enables precise, simple, and cost-effective solutions of genetic manipulation of stem cells to induce specific genetic alterations, unleashing its potential in both basic and applied sciences. Since CRISPR technology is only a few years old, it is reasonable to expect that more refined versions of Cas9 family proteins will provide even more fine-tuned approaches to genome engineering with fewer off-target effects, which will in turn facilitate the generation of edited patient-derived stem cells suited for personalized medical needs. So we will study more and more about the CRISPR-Cas9 and how stem cells are used in the coming classes. Thank you.